All right, so today we're going to talk about T cell effector function, and also kind of related to what I was just saying about the review session. The T cell effector function information will be the last information that's on the exam. So the stuff I talk about on Friday will not be on this exam unless they're like 2 or 3 slides I don't get through from this. Then those like last 2 or 3 slides will be exam. So it's basically the T cell effector function is where we're going to cut off. And so all of what we're thinking about here is regarding T cells in the periphery. And so remember that we have been in the secondary lymphoid organ where our naive T cell is getting activated. It's getting signal 1, getting signal 2. It's getting some cytokines like IL-2 production. It is undergoing some proliferation. And then it is differentiating to be able to have its effector function. That actual differentiation process does still happen in the secondary lymphoid organ, although we don't usually see the cell doing its effector function until that cell is out into some peripheral tissue. And so during the process that's shown on the left-hand part of this slide, you can imagine that lymphocyte, that T cell is undergoing lots of changes. And one of those changes, of course, is in trafficking molecules. So that at the beginning, that T cell was directed to the secondary lymphoid organ, is traveling as the red cell around to lymph nodes. That cell will eventually start making different trafficking molecules. It will be activated and become the blue cell so that it can actually go to some kind of site of infection or injury where it's going to do its effector function. And so in sort of this time, they're also where we're sort of differentiating and going to the effector function, that cell may also be moving towards the tissue. I left you off last time talking about the specifics of the differentiation for a CD8 T cell. And so a cell that starts out as a naive CD8 T cell, so it comes out of the thymus as a CD8 T cell, but does not yet have any effector function, can get activated and will become a cell called a CTL. CTL stands for cytotoxic T lymphocyte. And if you look at the word cytotoxic, toxic means it's going to kill something. And cyto means it's killing a cell, because cyto is for cell. And so our naive CD8 T cell is going to become activated to become a CTL. Finally, it's going to be able to do something about a microbe. And so you can see here is our naive CD8 T cell. It's getting a signal through MHC class 1 and peptide because it's a CD8 T cell. It's also getting signal 2 from CD28 on the T cell interacting with B7 on the APC. And that T cell is now going to proliferate and make many copies of itself, as well as differentiate to become a CTL. And now that CTL is going to be able to perform its effector function in the tissue. So here's that cell getting activated, ideally by a dendritic cell in a primary lymphoid organ. Here's that cell proliferating as well as differentiating. And now that cell has the ability to be a CTL in the tissue. And I happily actually have working videos today. So just to show you 2 examples of this. This is the one that I usually use. So the long cells are the long skinny cells are in this case, we'll just say that they're infected cells. They're actually tumor cells, but we're not going to deal with that right now. So we'll say these long skinny cells are infected cells. These smaller circular cells are T cells. And you're going to watch the T cells interact with the infected cells and then lead to the death of the infected cells. One thing to notice is that the T cell is able to interact multiple times. So the same T cell can actually kill multiple cells. But it's not like everybody in the area dies all at once. You can also notice that when this cell is dying and undergoing apoptosis, its membrane becomes compromised. And the way that you'll actually be able to see that here is that this experiment involves a dye in the media. So everything is clear normally, but if the dye gets into cells, then the cells change color. And so you'll be able to see the membrane does get compromised of this cell because it's going to change color. And so we can watch, maybe, possibly. Really? Really not going to work? All right. We may still not be able to watch this video, which is really sad because it's a good video. Happily, I put up a second one too. And so here we've got our infected cell, which is, as it says here, a cancer cell again, in blue. We've got our cytotoxic T lymphocyte here in purple, and we're going to watch that interaction here as well. And if I remember correctly, they do have a dye in this one as well that you can see when we're actually getting a signal transmitted to make that infected cell die. So you can see they actually will interact. That T cell basically puts something into the infected cell, and then you can see that infected cell undergoes apoptosis, and we completely lose that infected cell. Its membrane is entirely compromised. And so you can see basically there's something that the cytotoxic T cell put into this infected cell, and then the infected cell undergoes apoptosis. And so this is sort of that process of a CTL killing an infected cell. Yeah, Josh. Yeah.
Can cancer cells inhibit MHC?
Yes, when we talk about cancer immunology, we will discuss many of the ways that cancer cells inhibit these exact processes. That's one reason why I always find it hard. A lot of these videos, they show a cancer cell, and it's like, I don't want to talk about the cancer, like the specific cancer details yet, but yes, cancer cells can inhibit it. We will get to there with the cancer cell inhibition going forward. So, it'd be slideshow. No, I don't want weird. Just make it a slideshow. Thank you. I also don't want whatever this weird coach thing is. Make it go away. Just look like a normal thing. All right. And so here you can again see sort of your textbook's version of the CTL killing. So notice that the CTL is making specific contacts with one infected cell. It is inducing that infected cell to undergo apoptosis. The CTL is not harmed in this process. It can sort of detach and go find another cell to kill. But what you saw in the video was that the CTL was basically, secreting a packet or something to that infected cell to make it die. And no, it's not, going in 360 degrees, killing the whole tissue. It's a really specific interaction with that one infected cell, while the other types of cells, other cells that might be around that might not be infected, are not undergoing cell death. So I'm going to talk a little bit about sort of the mechanism of this in terms of specific molecules that are involved. When we think about killing by CTLs, we are absolutely thinking about apoptosis and apoptotic cell death. You saw it in the video as well. And so you saw that the cell will sort of shrink up and will make these little blebs, fragment the nucleus, and make sort of little pieces. You can see here's a healthy cell, here's an apoptotic cell, here's some of those membrane vesicles. So we're definitely seeing apoptosis happen in this situation. There are a couple different ways we can make that apoptosis happen. One of those methods of apoptosis is with a cell surface protein. So our T cell, which is shown here in purple, has a protein on its surface called Fas ligand. So FASL. FASL on CTL, see it even says the words right there. Which can interact with a protein called Fas. Probably not a shocker that the thing that interacts with Fas ligand is Fas. And that's on the target cell. So if the target cell has Fas, we can have Fas ligand on the CTL, induce apoptosis in that target cell. So basically, the CTL can specifically kill that target cell that has Fas on it. And you can see some of the details regarding Fas and Fas ligand here. So you can see Fas ligand on this cell with kind of like a pink membrane up here. That's the CD8 T cell. This is the infected cell that we're trying to kill. So it probably also had the MHC at some point that it was presenting. You can see it has Fas on it. And you can notice that there are a bunch of proteins that are involved downstream of Fas signaling. The key thing I want you to notice is that when we activate Fas, we turn on this caspase called caspase 8. And caspase 8 cleaves a lot of stuff in the cell. One of the things that caspase 8 can do is it can mess with the mitochondria. So we get all of the parts of mitochondrial apoptosis that you guys learn about in Bio 250. But the other big thing when we think about apoptosis is that whenever apoptosis happens, we have to turn on this enzyme called caspase 3. Caspase 3 starts out as an inactive protein called procaspase 3, and it gets turned on by cleavage. That can happen because of all this mitochondrial business. But one thing that's also really nice is that caspase 8 can just directly cleave caspase 3, get rid of all these middlemen, turn on caspase 3, and caspase 3 does things like cut up the DNA, cut up all the cell structures, destroy the cell, give us apoptosis. And so the idea that I want you to see here is that when we have Fas get triggered on an infected cell, we can get a pretty direct route to caspase 3 and apoptosis. Sometimes in some other cell biology situations, you need to have a whole bunch of steps before you can get all the way down here to caspase 3. Yeah, we do some of those other steps here, but we can also just have this really direct route where caspase 8, caspase 3, and we're done. So it can be a pretty quick apoptosis like you saw in the video. But in the video, it also is a little bit easier to see that the other thing that CTLs do is that they will have some granules inside of the cell. So once that cell has differentiated, once that cell has grown up to be an actual CTL, it's going to be full of these granules, which are referred to as cytotoxic granules. This image from your textbook, for whatever reason, is calling them LG or lytic granules. They're cytotoxic. They're toxic to cells. And in fact, they're toxic to cells because they lyse them. So it's lytic or it's cytotoxic. I usually think of them as cytotoxic granules. And in this image that you can see in the microscopy on the right, the cytotoxic granules are labeled in red. And so you can see in this CTL that is not yet killing, they're just sort of scattered around the cell. If that CTL actually interacts, it's now seeing MHC on a tissue cell. It now needs to kill this cell. You can see that the cell the T cell rearranges where its lytic granules are and puts them right there next to the contact point with the infected cell, and then actually secretes those lytic granules right there in that contact point so that they go to the target cell, the infected cell we're trying to kill, and they don't like float around and kill and potentially impact other cells. So it's sort of like this very specific secretion just in this little area where this cytotoxic T lymphocyte has this sort of poison stuff, has this killing stuff, and it secretes it directly just to this one cell that saw the MHC from. It's actually kind of cool how the signaling works. It turns out that there are basically a whole bunch of adhesion molecules like integrins actually holding the cells together, and they also make like a wall so that these lytic killing molecules can't like diffuse away. So like basically like the cells wall off the area and then shoot toxic stuff to kill their neighbors, which then will lead to that target cell undergoing apoptosis. You can see the same idea here. So here we've got our CTL. It's full of some pink dots, which are the cytotoxic granules. As that CTL interacts with the target cell, we're now going to move those cytotoxic granules right next to the target cell. And then we're going to secrete the toxic stuff to kill that target cell. Now the T cell can actually leave, and it can go kill somebody else if it needs to kill another infected cell. The infected cell is going to undergo apoptosis. And so you can see this happening here. What's nice about this is that it also does give sort of a timeline of how long some of these steps will take, which I know sometimes people find interesting. So when people think about CTL killing, they very often think about cytotoxic granule release. When we think about that cytotoxic granule release, we can also say, well, what's in the cytotoxic granules? What is the toxic stuff? Immunologists have described a lot of things that are in the cytotoxic granule. I am going to tell you the names of two things that are in the cytotoxic granule, though in reality, there's a ton. I could go on and on about all sorts of different proteins that are inside that cytotoxic granule, but there's two key proteins that I'm going to mention. One of those proteins is called perforin. You can see perforin at the top of this slide. When you hear the word perforin, what does it sound like to you? What does it sound like, Brenna? How about perforate? What does perforate mean? What does it mean, Brooke? That's a great, Brooke has like a great hand gesture. Huh? Puncture, like poke holes in is what it means. Perforate means pokes holes in. Technically, this is perforated paper because it has holes in it. Perforate means poke holes in. Guess what perforin does? It pokes holes in cells. And so perforin is in that cytotoxic granule. It gets secreted by our T cell towards the target cell. And you can see that that perforin molecule makes holes in the target cell. So we make these perforin pores in the target cell. Now, things that are supposed to be inside the cell leak out. Things that are supposed to be outside the cell leak in. We might have osmotic shock. We can have all sorts of things. Basically, we've killed our cell because we've perforated it. And you can see an image of perforin pores in a target cell on the right. If you look at this electron microscopy image of some of these perforin pores, so you're basically looking at these little pores that you see in the cartoon on an actual cell here. Is there anything that you notice when you look at that image? Or anything that image reminds you of from earlier in the semester? Complement. That looks a lot like the membrane attack complex and C9. the pore that complement makes. In fact, they are almost indistinguishable. And I always sort of laugh when I look at an immunology textbook because I cannot tell you if this is a complement, if this is really complement or perforin. They say it's perforin, but I think sometimes they actually lie. But that's okay. Because they look identical. And so it's the same kind of idea. We're poking holes in something to get rid of it. What is it's just a triangle pointing to one of the perforin pores.
Something else
No, it's just trying to point to one of the perforin pores, though I can't imagine why you need someone to point to it because they're kind of obvious. There are, as I said, other things in this lytic granule or in this cytotoxic granule as well. And the one other one besides perforin that I'm going to tell you about is one that's called granzyme B. And one thing to note, if we look at the name granzyme comes from the fact that this is an enzyme that's in the granule. So it's a granule enzyme. And so it must have some enzymatic activity. And granzyme B does have some enzymatic activity. It's this little purple oval. Granzyme B, just like we saw with some of the other caspases with FAS, can sort of get rid of the middleman, activate caspase 3, and get us to the end of apoptosis, and sort of just cut right to the chase, get to the end of the apoptotic cascade because it's directly cleaving caspase 3. It can also turn on some of the other parts of the apoptotic cascade, but really it's sort of a direct route to caspase 3 as well. One thing that is also discussed often is one thing that's really nice about the fact that we have this vesicle that has both granzyme B and perforin in it. So you can see up at the top in our T cell, we've got granzyme B and perforin. When we make a perforin pore, that also is a nice way for granzyme B to get in. So the perforin pore can allow granzyme B in. It can also actually directly be part of lysing the cell. And so some people debate how much it's about lysis and how much it's just about letting granzyme B in. In reality, the answer is it's doing both, but some people will talk about all that. Yep, Jonah.
So then is granzyme B and FAS-L both factors when it comes to just getting caspase 3?
They both are getting us right to caspase 3. Sometimes, the FAS will be more important. Sometimes the granzyme B will be more important. That's a T cell specific problem. But they're just two of the key things we think about with cytotoxic T cell killing. And you can see that exact sort of thing on this slide as well. We often will see sort of the FAS ligand on the T cell with FAS, as well as things like perforin and granzymes being made by our cytotoxic T cells. And so we've got perforin, we've got granzymes, we've got FAS and FAS ligand. The one other reason why I show you this slide is that sometimes we also see some cytokine production by cytotoxic T cells or CTLs. The important cytokine that I often think about with CTLs is called interferon gamma. And so we can sometimes also see some interferon gamma being produced by CTLs. Your book in some cases talks about IL-2. I want you to completely ignore that. As far as we're concerned, all T cells make IL-2 because I don't want to get into the weeds there. So we'll just say all T cells make IL-2. The thing that's unique about the CTLs is that they can also make interferon gamma. So that was sort of the bottom part of this slide and the effector functions we see with a CD8 T cell. But we can also think about the CD4 T cell and its differentiation process. So what functions does the CD4 T cell gain? And what does our effector CD4 T cell do? Very big picture with a CD4 T cell. That CD4 T cell will sometimes be called a helper T cell. So just like with our CD8 T cell, it starts out as a naive CD8 T cell and becomes a CTL. With the CD4, it starts out as a naive CD4 cell and becomes a helper T cell. Oftentimes we'll write like a T with a little H next to it if we mean an activated CD4 cell because it's a helper T cell. And so you can see I already did that on this slide title, that it's a helper T cell. I've got that T sub H. And the key thing about a helper T cell, when that helper T cell is helping, is that the helper T cell is going to secrete a lot of cytokines. The secretion of those cytokines in a lot of ways is really similar to what the CTL is doing in secreting its lytic granules. It's just now it's not a granule that's going to kill something. It's a granule full of cytokines that's going to help because it's a helper cell. But it's the same thing, like we have the cell kind of rearrange and put the granules right next to the cell that it's going to secrete towards, and there's even the same kind of wall with the integrins, and we get the same little directional secretion. But what you might notice as I discuss this is I just said that our helper T cells secrete cytokines. I didn't tell you which cytokine was secreted. The reason for that is that there are multiple types of helper T cells. So there are many different helper T cells that will make different cytokines. And so we have to kind of think about each of the different types of helper T cells and those cytokines here. When the T cell comes out of the thymus, it is not one of these types. It's just a plain old CD4 T cell. However, when that CD4 T cell is getting activated, when it's going through this activation process, it is going to get a signal that tells it which kind of helper T cell it needs to become. And that's going to be based on what kind of cytokines that helper T cell receives in this process. So when we get to the effector CD4 T cell, it's going to be one of these special kinds. And so I'm going to tell you all these special kinds of helper T cells. It wasn't that special kind coming out of the thymus in most cases, and really became that special kind of helper T cell based on the signals it saw during its differentiation. And so you can see this sort of related to what we were talking about before here. So with a CD4 T cell, that CD4 T cell starts out as a naive T cell. It's going to get its signal 1, it's going to get its signal 2, but it's also going to get some kind of cytokine. This is where that signal 3 becomes really important, some other cytokine signal from that antigen presenting cell. And that is going to change what's going on in that helper T cell. It's going to help it differentiate to become an actual helper T cell instead of just a naive CD4. But it's also going to tell it what kind of helper T cell it should be. And so when we talk about helper T cells, we're going to talk about subsets of helper T cells. And so we're going to have to think a little bit about what are these different subsets. There are some times where some immunologists can get a little over the top with their helper T cell subsets. I swear, sometimes I feel like I read a thing and it's like, we have discovered the T cell subset that fights against fungal infection in your right pinky finger on Tuesdays. Like, they're so specific, and sometimes it's a little much. So when we think about helper T cell subsets, I'm not going to tell you every one that's ever been proposed. And one of the things that we're going to be thinking about when we think about our helper T cell subset is that in fact, for me to think it's a real helper T cell subset, there are certain sort of questions about it that have to be answered. And so we're going to be thinking about those questions for each of our helper T cell subsets. And so one of the things that we're going to have to think about, so the blue is our T cell that is becoming a helper T cell and becoming one of these types of helper T cell subsets, is what cytokine does the cell have to get during activation in order to tell it to become that subset? And so that's sometimes referred to as a polarizing cytokine. So it's somehow that T cell is going to receive some signal, some polarizing cytokine that is going to tell it become this special type of helper T cell. When that polarizing cytokine is received, that changes that naive T cell. And one of the things that happens, there's tons of things that happen to the naive T cell, but one of them is a specific transcription factor gets made. And that tells this T cell to do all the things of whatever subset it is now declaring its major to be. And so one of the things in my mind that like makes you a legit helper T cell subset is if you have a specific transcription factor that we can name. And so what we know is that there's some polarizing cytokines that turn on some transcription factor, which this says is the master gene regulator. Once our cell starts making that transcription factor or master gene regulator, now it starts doing its own special things of whatever that subset does. And that would be what kind of cytokine this helper T cell produces. And so because the cell is making its transcription factor, that will tell the cell, you should make this effector cytokine. Some of the effector cytokines and some of the outputs for helper T cell types are really good at responding to viruses. There's another one that's really good at helping the cell respond to, or helping the body respond to fungi that are extracellular fungi. There's another one that's really good at helping this body respond to parasitic worms. There's sort of lots of different effector cytokines that might be particularly useful in a certain type of pathogen situation. And so ideally, if that pathogen is around, what we want to have happen, what we as the organism want to have happen, is we would like our T cells, so if I have a fungal infection, I would like to turn on the type of helper T cell that's really good at getting rid of fungal infections. I don't want to turn on the parasitic worm kind. I want to turn on the fungal infection kind. And the way I would do that is I would make some polarizing cytokine to say, hey, T cell, become the fungal infection kind. And then the T cell is going to grow up and become the fungal infection kind. That polarizing cytokine is generally coming from, say, that first infected cell, that first cell that's presenting on MHC. How do you think that cell might know that it wants a fungal infection T cell or a parasitic worm infection T cell or a virus infection T cell? How will that cell know which direction it should push its T cells in? Yeah, Brenna. Okay, what else might be going on when it obtains that peptide? Yeah, Brian. IRF. What are those? Where do those come from? The NF-kappa B and the IRF. Look at the top. Yeah, remember PRRs? And the PRRs can kind of relate to a different kind of response. Whatever PRR this cell saw is going to change what cytokine it makes. And that cytokine is going to change which direction we push the T cell in. And so you can see that here as well. So you can see, if we have a bacteria hitting some bacterial PRR, then that's going to make our cell make one kind of cytokine. and that will push our T cell in one direction, which, oh my gosh, is the good kind for bacteria. If we have a parasitic worm, it's going to tell the cell to make a different cytokine, and that's going to push the T cell in a different direction to be the kind of T cell that's good for parasitic worms. And so there's this real sort of interaction, again, between that, what the innate immune response type was, and how that directs the type of adaptive immune response. Sometimes people talk about some of these T cells as like the quarterbacks of the immune system, of directing the immune response in different directions. If that's the case, then the PRRs are like the coach who told the quarterback what direction to throw things in. And so the innate immune response is very much directing which direction these T cell subsets are going in. So this is one view of the T cell subsets. This one is not from your textbook. It's from a different textbook. I like it because it has sort of the information about effector functions written out nicely here. And the similar table in your textbook doesn't have this quite as clearly. And so that's why I'm using this. This image shows more kinds of helper T cells than I am going to tell you about. So I am not going to tell you about all of the ones in this image, but I like this image because it has these effector functions described out for us really nicely. This is an image from your textbook. And these, in fact, are the helper T cell subsets that we're going to talk about. What you can notice in this table, and you'll probably want to look at this table in prepping for the exam, is that we have this part right here that says cytokines that induce differentiation. We could also call that the polarizing cytokine. And you can see that in some of these cases, we've got a couple things listed. For all but one of these subsets, I'm really going to mention one cytokine as being key. For one of them, I got to say two, but the rest of them, I'm just going to say one cytokine. Then you can see the transcription factor that's important for that cell type, and those transcription factors are also listed here. And then you can see the cytokine that is produced by the subset down here. Again, you can see that there are often multiple cytokines that are listed. I'm usually going to mention one of them. I will also tell you, when I am grading anything related to this on an exam, while I'm going to focus on one cytokine for this scenario and this scenario, if you write one of the other ones that's in your textbook, so like I'm going to talk about IL-4, but if you just write IL-5 for this one, you get credit. So note that like this is what I'm going to go with as like sort of what the correct answers are, even though I'm going to focus on one particular cytokine in some of these situations. This whole idea of thinking about helper T cell subsets was first described when somebody came up with the idea that maybe there were two kinds. And so at first, we just thought there were two kinds of helper T cell subsets. Actually, when I went to graduate school, when I started graduate school, there were two kinds. When I ended graduate school, there were like 20. It was great. So these were the two when I started graduate school. And they are called either Th1, helper T. We know it's a helper T cell. We know it's a CD4 T cell because it has Th in it and the type number 1, or Th2. And so originally we thought all of our helper T cells were either Th1 or Th2. We now know they've got some other choices, but this is sort of the original idea. And with Th1 cells, as you can see here, we have a cytokine called IL-12 that is made by our innate immune cell, usually our dendritic cell. That turns on the transcription factor. It tells that naive T cell to start making a transcription factor known as TBET. And then the effector cytokine that will be made by the Th1 cell is interferon gamma. And alternatively, the idea was there was also Th2. Th2 was polarized by IL-4. I will say that now we think that another cytokine called IL-33 is important there. We make this transcription factor called GATA3, and now the Th2 cells make IL-4. And the reason why I point out the IL-33 up here is because people usually get really angry. They're like, but it needs IL-4 to make IL-4, and they get really annoyed. It's probably IL-33, and in reality, it's making IL-4, IL-5, and IL-13. But if you just know IL-4 polarizes, IL-4 is made, that's fine. But if that stresses you out that it's the same cytokine, there's some other options for you. And you can see this sort of Th1, the same sort of Th1 situation on this slide. So in general, with our Th1, we're going to polarize that naive T cell by giving it IL-12 when we first activate it. When that cell is first activated, it's going to start making this transcription factor TBET, and that is going to tell that T cell that it should be a helper T cell who helps by making interferon gamma. So each of our helper T cell subsets help by making a different cytokine. In this case, it's interferon gamma. Interferon gamma, and so we need to think about, well, who cares about interferon gamma? What's interferon gamma good for? What's it useful for? One of the things that interferon gamma can do really well is it can improve antigen presentation and activate antigen presenting cells. Interferon gamma can also turn on CD8 T cells. And interferon gamma can do one other function that I'll show you on the next slide, which is activate macrophages. So interferon gamma is really great for turning on, like really making MHC presentation awesome, really making CD8 T cells work well as activating macrophages. What kind of pathogen is going to be helped when you have a lot of CD8 T cells getting activated? So if we like enhance our CD8 T cells, what pathogen are we going to be really good at dealing with? Yeah, a virus. With the macrophage activation, remember that we've talked about the fact that macrophages can phagocytose stuff. Sometimes that stuff can kind of hang out in the phagosome, and maybe the macrophage isn't going to be really good at killing it. The other thing that interferon gamma can do is that interferon gamma can activate the macrophage, so the macrophage is really good at killing anything inside it. So now that macrophage is going to be making way more of the things that help it degrade microbes. So if there's anything in that macrophage, we are going to destroy the heck out of it. And so in the end, the Th1 response is really good against sort of viral infection. It's also really good against bacteria that are inside of cells, intracellular bacteria. All of our different helper T cell subsets can go wrong sometimes. When a Th1 response goes wrong, sometimes we can get some autoimmune diseases as well. And so sort of if it's wrong, autoimmune disease. If it's right, we're dealing with viruses and intracellular bacteria. And so we're going to, with our Th1 cell, we're going to activate macrophages and CTLs, and we're going to get rid of intracellular bacteria and viruses. And that's really all because of the actions of interferon gamma. The other one of the sort of big original T cell subsets what is the Th2 subset. The Th2 subset is polarized by IL-4. Thus, that starts the cell making the transcription factor GATA3. And now Th2 cells will start making a bunch of other cytokines called especially IL-4. If you sort of read a bunch of immunology literature, sometimes people refer to these responses also as just type 1 and type 2 responses, because your immune system ends up going in like totally different directions in these. And so I mention this because something that my sister is currently working on involves type 2 responses. And so she's always like, if you read good type 2 papers, let me know. So this would be type 2. So just like with the Th1 response, we need to think a little bit about what IL-4, 5, and 13 do for us. Like, how do they help us? What kind of response do they give us? And so I'm going to show you on the next slide some of the big things that we think about some of these Th2 cytokines doing for us. And so one thing that can happen is, and I'll say more about this later, it has to do with antibodies. We'll get there in a second. But a big thing when I think about Th2 cells is I think about things like mast cell degradation. So we're going to activate mast cells pretty massively with our Th2 cytokines. We're also going to induce a lot of mucus secretion, and this says peristalsis because it's talking about the GI tract, but to be honest, it's really mucus secretion and expelling stuff. So if I was in not the GI tract, if I was in the respiratory tract, it would be like sneezing. It would be trying to make things go out, trying to like throw things out. And we also see eosinophil activation. And so usually with when we think about the Th2 cytokines, we're thinking a lot about mast cell activation. And I often think about trying to expel stuff. Remember, when we think about mast cell activation, mast cells are full of granules. They are a tissue type of granulocyte. And the idea with a mast cell is that we've got all of these toxic granules. It's not the same stuff that's in CTLs, unfortunately. It's different toxic stuff. But the idea is that a mast cell will secrete that toxic stuff at an extracellular pathogen, a pathogen that's outside of cells. Ideally, in theory, a pathogen that's too big to be phagocytosed. So if you remember back to lab 1 where you saw things like those trypanosome parasites outside of cells, those were kind of big. The idea is we're shooting toxic stuff at them to try to kill them. And so with our Th2 response, we're generally thinking about some extracellular pathogen that we're trying to kill, either by shooting toxic stuff at it from our mast cell or just by trying to remove everything from our GI tract or remove everything from our respiratory tract to just like throw it out. We're trying to expel it and get rid of that extracellular pathogen however we can. And so you can see that we think a lot about Th2 cells as being really important for getting rid of extracellular pathogens. So I don't know how to spell anymore. Pathogens. Parasitic worms, like helminth worms, are some of the most famous pathogens that we think about with Th2 responses. Th2 responses also, like any of these other kinds of responses, can go wrong. And when a Th2 response goes wrong, you end up with allergies. It is clear for a lot of reasons that my body is predisposed to make Th2 responses very frequently, and that's why I'm allergic to life. But you can see that, especially kind of, and so you can see, I guess, this one. Here is kind of that overall idea of the Th1 versus Th2 control. So if our innate immune cell maybe sees a virus, that's going to tell that innate immune cell, hey, maybe you should make some IL-12 while you're presenting. So we're going to make our helper T cell become a Th1 cell, and now it's going to make interferon gamma, which is really good for dealing with viruses. And then we all live happily ever after. We started with a parasitic worm. That might tell our dendritic cell to make IL-4. So our naive T cell might get pushed in the direction of becoming a Th2 cell. We might make some more cytokines like IL-4. They're going to help us get rid of that worm. One other thing that we have noticed here is that a lot of these cytokines also really control class switching. And so if we think about cytokines like interferon gamma, which is made by a Th1 cell, you can see that interferon gamma tends to induce class switching to things like IgG3 and IgG2A. Those are really good at dealing with viruses. So again, we're getting class switch towards antibodies that are particularly useful in this situation. If we look at IL-4, our key cytokine for Th2 cells, you can see that the most famous thing that happens with IL-4 is that we induce IgE, so class switch to IgE, which is the one that goes with mast cells and the one that is actually involved in dealing with parasites. So not only are we kind of changing how various cells are working that you can see here, we're also going to be impacting the B cells and changing which type of antibodies are being made, because we're going to make the antibodies that are most useful for any particular situation. One of the other things that comes up on this slide also is something that comes up more on some later slides, but you can also notice with our interferon gamma, we can see induces some types of antibody class switches are induced, but you can also see some are inhibited. And so you can see with that Th1 cytokine, we're inhibiting IgE, that more Th2 kind of isotype. And that's a really key thing to understand. There is a lot of feedback between Th1 and Th2 cells. So I've told you that if our T cell gets IL-12, that T cell will start making TBAT, will start making interferon gamma. But there's actually a little more going on than that. Once we have that IL-12 and once we have that TBAT made, TBAT is actually going to turn off GATA3. TBAT is going to turn off IL-4 and IL-5. If we start making a Th2 response, then we start making GATA3, GATA3 is going to inhibit TBAT. GATA3 is going to turn on IL-4 and IL-5, but it's also going to inhibit interferon gamma. And so each of these types of responses sort of gives a positive feedback loop to get more of its same response and actively turns off the other kind of response. It's sort of like whenever you start making a T cell response, you have to make Th1 or Th2. You can't really make both. And so you start making Th1, we're going to turn off. Nobody in that lymph node is going to make Th2. Everybody in that lymph node is going to do Th1, and we're just going to have like a Th1 positive feedback loop. If somebody starts making a Th2, we're going to get a Th2 positive feedback loop, and we're going to turn off all the Th1s around. This is actually something that's really interesting and that we'll see more about a little bit later, where sometimes we can actually try to flip the balance of a response and actually change some pathology, and that's something that people are starting to think about a bunch right So this is where I was where things were when I finished graduate school, or when I started graduate school. By the time I finished, we had some more subsets. And sometimes people don't like the other subsets because they aren't like numbered number 1, number 2. You're like, did you immunologists forget how to count? I mean, I know you didn't know how to count with complement, but now you forgot how to count again. But it turns out the names they have are actually kind of useful. So the next T cell subset that I'm going to tell you about is a T cell subset that's called a Th17 cell. To give you the spoiler to start the whole thing out, the reason why a Th17 cell got named a Th17 cell is because its effector cytokine, the cytokine that makes it special, is IL-17. So that's how it got the name Th17. Th17 cells make a transcription factor called War gamma T. And this is the one situation where I'm going to point out 2 cytokines for polarizing, and having the 2 cytokines for polarizing here is important. And a bunch of them are listed, but what I'm going to list for you is TGF beta plus IL-6. And I'm picking IL-6 actually on purpose here. So have you seen IL-6 before? The answer is yes, because I asked that question, and because I can like randomly ask you that question. The answer is no, I guess. So where have you seen IL-6 before? Maybe, yeah. It was part of that innate immune inflammation process. It's one of the key inflammation cytokines. And so what I want you to notice with Th17 is we need this cytokine called TGF beta plus inflammation. So we need some inflammation to tell a T cell to become a Th17 cell. And so this is the one where I give you 2 cytokines, because the idea is you need TGF beta plus inflammation to make this happen. And when we see Th17 cells made, those Th17 cells we now know are really good extracellular bacteria. They're dealing with extracellular bacteria and fungi. And one of the reasons why they do that is that they activate neutrophils. So we're sort of going to enhance neutrophils' ability to do their job, and we're going to go get any extracellular bacteria or fungi. There are also lots of situations where if we make Th17 cells inappropriately, we end up with inflammation or autoimmunity. One other type that I want to mention here is was known as the TFH cell. The name TFH is actually really important in telling us some things about what the cell does. So it actually gets sort of down here in what's going on here. The TFH, the FH stands for follicular helper. So it helps in a specific place called the follicle. It's a follicular helper. And specifically, it helps, let's do it this way. And specifically, it helps B cells in follicle. So T follicular helper cells are really key for helping us make a good B cell response. If you remember when we talked about B cells, we kind of left them as they were coming out of the bone marrow. We didn't talk about their effector functions. We didn't talk a ton about what they were doing in the periphery. And the reason is because they don't really do anything in the periphery without TFH cells. So I had to tell you about TFH cells before I can tell you anything else about those B cells in the periphery. We'll give our polarizing cytokine for TFH cells as IL-21, the transcription factor as BCL6, and the cytokine, the effector cytokine as IL-21. And so if we really want to have a good B cell response, we need T follicular helper cells. And so here you can kind of see this indicated here. Here's our B cell. It's going to, in order for it to really be turned on, it's going to need some cytokines from a T follicular helper cell. And so in terms of material that will not be on the exam, you'll see more about B cells. That's a later problem. But I have to tell you about the fifth, final, and in many ways coolest, or very cool type of T cell subset. And these cells are known as the Treg cells. There are two different ways that we can think about regulatory T cells, or Tregs. And before I tell you that, I want to tell you there's a whole long story that I could do that I'm not going to give you time for right now. But I will tell you that once upon a time, these were called Th3s, but Treg is a better name. So we did know how to count, I promise. Once upon a time, these were actually discovered in like the 80s. And then there were some experiments done that didn't support them. And people decided they didn't believe in them anymore. And then in the late 90s, early 2000s, we were like, oh, wait, no, they really are a thing. But they had a different name in the 80s. For whatever reason, it's like immunologists hate that word. And so we never say that word, even though it's a really useful word in this situation. So if you see regulatory, or if you ever hear an immunologist say regulate, the word they really want to say is suppress. But we don't use suppress anymore. Suppress is a dirty word in immunology. And so the idea with a regulatory T cell used to be called a suppressor T cell, but we don't say suppressor T cells anymore. I did not get struck by lightning. Is that regulatory T cells turn everybody else off. Regulatory T cells are the off T cells that will turn off their neighbors. And so here, your textbook, I guess, is daring. It actually says the Treg is suppressing its neighbor cell. All Tregs make this transcription factor that's called FOXP3. Tregs do their suppression largely by producing a cytokine called IL-10. And there are actually two ways we can make Tregs. One of the ways that we can make Tregs is by giving those Tregs some TGF beta. And so we can provide TGF beta. And so now you'll notice it's TGF beta without inflammation, as opposed to before it was TGF beta with inflammation. And now we're going to tell this T cell, you need to be an off switch. You need to shut everybody else down to keep us from getting immunopathology. And so you can see that these cells are going to be inhibiting inflammation and really just inhibiting all of your other immune responses. The other thing that's important to know about Tregs is that, as I just said, there are two ways you can make Tregs. One of them is through this process that's shown here, where we polarize a naive T cell with TGF beta, we tell it to turn on FOXP3, and then we start making IL-10. Some Tregs actually come out of the thymus as Tregs. And so these are the cells. If you remember when we talked about T cell development in the thymus, I told you there were 4 choices. There was high signal, we kill it. No signal, neglect. Lowish signal, positive selection. And I said there's kind of a medium high, but I'm not going to talk about it. This is the medium high. If a cell gets a medium high signal in the thymus, then that cell is going to start making FOXP3. So it's just a different way to get FOXP3. And that T cell is going to come out of the thymus as a Treg. Because these are the ones that had kind of the second highest signal, they're kind of dangerous. And so we're going to let them only be Tregs and turn things off. We're not going to let them actually induce different types of responses. So this is a slide you saw before. And what you can notice is now I just don't have a white box. over that part where we develop Tregs out of the thymus. So there's 2 ways to make Tregs out of the thymus or not. You can see that Tregs can suppress by making inhibitory cytokines like IL-10, so they can actively suppress other things. They also have tons of IL-2 receptor, so sometimes they just steal all the IL-2, so nobody else can have the IL-2. So sometimes they're working through cytokine deprivation. And just like when we originally had this idea that like Th1s inhibited Th2s and Th2s inhibited Th1s, now that we know about all the different kinds of helper T cell subsets, we know that they all inhibit each other. So if you make a Th2, it inhibits Th1 and it also inhibits Th17. Th1 inhibits Th17 and Th2. And so it's the same kind of idea where once you pick one of these subsets, you're sort of going to drive your whole immune response. We'll say it for that day or for that situation. Everybody's going to go in that direction, and we're going to inhibit other directions, maybe in that lymph node or for that short period of time. And so there's a lot of feedback between all of these subsets. We tend to push responses in one direction or another. Okay, that's everything I wanted to get through before the exam. The stuff I talk about on Friday will not be on the exam. Here's my summary of T cell subsets. It's also in a table. And I will see you guys tomorrow in lab. The lab handout is already posted.